10th Standard Science, Chapter 18, Heredity. இந்த லெசனோட புக் பேக் ஆன்சர்ஸ் பார்ப்போம். First Roman letter, choose the correct answer. First one, according to mental alleles, have the following characters. Option B answer, responsible for character. Second one, 9:3:3:1 ratio is due to option C answer, independent assortment. Third one, the region of the chromosome where the spindle fiber get attached during cell division, option C answer, centromere. Fourth one, the centromere is found at the center of the dash chromosome, option B metacentric. Fifth one, the dash units form the backbone of the DNA, option D answer, sugar phosphate. Sixth one, Okaski fragments are joined together by dash, option D answer, DNA ligase. Seventh one, the number of chromosomes found in human beings are dash, option A answer, 22 pairs of autosomes and 1 pair of allosomes. Eighth one, the loss of one or more chromosome in a ploidy is called dash, option B answer, aneuploidy. Second Roman letter, fill in the blanks. First one, the pair of contrasting character traits of mental are called dash, alleles. Second one, physical expression of a gene is called phenotype. Third one, the thin thread-like structures found in the nucleus of each cell are called dash, chromosomes. Fourth one, DNA consists of 2 dash chains, polynucleotide chains. Fifth one, an inheritable change in the amount or the structure of a gene or a chromosome is called dash, mutation. Third Roman letter, identify whether the statement are true or false. Correct the statement. First one, a typical Mendelian dihybrid ratio of F2 generation is 3:1. The statement is false. The correct statement, a typical Mendelian dihybrid ratio of F2 generation is 9:3:3:1. Second one, a recessive factor is alterated by the presence of a dominant factor. The statement is false. The expression of a recessive factor is alterated by the presence of a dominant factor. Third one, each gamete has only one allele of a gene. The statement is true. Fourth one, hybrid is an offspring from a cross between genetically different parent. The statement is true. Fifth one, some of the chromosomes have an elongated knob-like appendage known as telomere. The statement is false. The correct statement, some of the chromosomes have an elongated knob-like appendage known as satellite. Sixth question, new nucleotide are added and new complementary strand of DNA is formed with the help of enzyme DNA polymerase. The statement is true. Seventh one, Down syndrome is the genetic condition with 45 chromosomes. The statement is false. Down syndrome is the genetic condition with 47 chromosomes. Fourth Roman letter, match the following. Answer, first one, autosomes. Answer, 22 pairs of chromosome. Second one, diploid condition. Answer, 2n. Third one, allosome. Answer, 23rd pair of chromosome. Fourth one, Down syndrome. Answer, trisomy 21. Fifth one, dihybrid ratio. Answer, 9:3:3:1. Fifth Roman letter, answer in a sentence. First one, what is a cross in which inheritance of 2 pair of contrasting characters are studied? Answer, dihybrid cross. Second one, name the conditions when both the alleles are identical. Answer, homozygous condition. Third question, a garden pea plant produces axial white flower. Another of the same species produced terminal violet flower. Identify the dominant trait. Answer, position of flowers. Axillary position is dominant over terminal position of flowers. Next one, color of flowers. White color is dominant over violet color. Fourth question, what is the name given to the segments of DNA which are responsible for the inheritance of a particular character? Answer, genus. Fifth question, name the bond which binds the nucleotides in a DNA. Answer, hydrogen bonds. Sixth Roman letter, short answer questions. First one, why did Mendel select pea plant for his experiments? First point, it is naturally self-pollinating and so is very easy to raise pure breeding individuals. Second point, it has a short lifespan as it is an annual and so it was possible to follow several generations. Third point, it is easy to cross-pollinate. Fourth point, it has deeply defined contrasting characters. Fifth point, the flowers are bisexual. Second one, what do you understand by the term phenotype and genotype? External expression of a particular trait is known as phenotype. A genotype is the genetic expression of an organism. Third question, what are allosomes? First point, allosomes are the chromosomes which are responsible for determining the sex of an individual. Second point, they are also called as sex chromosomes or heterochromosomes. Third point, there are two types of sex chromosomes, X and Y chromosomes. Fourth question, what are Cascade fragments? Answer, during the replication of the DNA molecule, the new strand is synthesized in short segments which are called Cascade fragments that are joined together by the enzyme DNA ligase. Fifth question, why did euploidity considered to be the advantageous to both the plants and animals? Answer, the euploided plants often result in increased fruit and flower size. Therefore, it is advantageous for them. Second point, the euploid animals are sterile. Sixth question, a pure tall plant TT is crossed with pure dwarf plant TT. What would be the F1 and F2 generations? Explain. Crosses involving inheritance only one pair of contrasting characters are called monohybrid cross. Cross between pure tall TT plant and pure dwarf TT plant. Parental generation. Pure breeding tall and dwarf plants are crossed TT into TT. F1 generation. Monohybrids are heterozygous tall. F2 generation. Selfing of the F1 monohybrids takes place. Tall and dwarf plants are obtained in the ratio of 3:1, which is the phenotypic ratio. Genotypically, plants are of three types as shown above and therefore genotypic ratio is 1:2:1. Seventh question, explain the structure of a chromosome. Answer, first point, the chromosomes are thin, long and thread-like structures consist of two identical strands called sister chromatids. Second point, they are held together by the centromere. Third point, each chromatid is made up of spiritually coiled thin structure called chromomina. The chromomina has a number of bead-like structures along its length which are called chromomeres. Fourth point, the chromosomes are made up of DNA, RNA, chromosomal proteins, histones and non-histones, and certain metallic ions. These proteins provide structural support of the chromosome. A chromosome consists of the following regions. Primary constrictions. The two arms of chromosome meet at a point called primary primary constriction or a centromere. The centromere is the region where the spindle fibers attach to chromosomes during cell division. Next one, secondary constriction. Apart from the primary constriction, some chromosomes possess secondary constriction at any point of chromosome. They are known as nucleus zone or nuclear organizer. Formation of nucleus in the nucleus. Next one, telomere. The end of the chromosome is called telomere. It maintains and provides stability to the chromosomes. Next one, satellite. Some of the chromosomes have an elongated knob-like appendage at one end of the chromosome known as satellite. The chromosomes with satellite are called as the shared chromosomes. Eighth question. Label the parts of the DNA in the diagram given below. Explain the structure briefly. Watson and Crick model of DNA. First point, DNA molecule consists of two polynucleotide chains. Second point, these chains form a double helix structure with two strands which run anti-parallel to one another. Third point, nitrogenous bases in the center are linked to sugar phosphate units which form the backbone of the DNA. Fourth point, pairing between the nitrogenous base is very specific and is always between purine and pyrimidine linked by the hydrogen bonds. Next one, adenine. A link thymine T with two hydrogen bonds A is equal to T. Then cytosine. C links guanine G with a 3 hydrogen bond equal to G. C is equal to G. This is called complementary base pairing. Next, fifth point. Hydrogen bonds between the nitrogenous bases make the DNA molecule stable. Sixth point. Each turn of the double helix is 34A degree 3.4 nm. They are 10 base pairs in a complete. Then seventh point, the nucleoids in a helix are joined together by phosphodiester bonds. Draw this picture and label the parts nucleotides in a DNA. Nucleotides in a DNA: P phosphate, A adenine, G guanine, T thymine, C cytosine. Seventh Roman letter, long answer questions. First one, explain with an example the inheritance of dihybrid cross. How is it different from monohybrid cross? Dihybrid cross involves the inheritance of two pair of contrasting characteristics or contrasting traits at the same time. The two pairs of contrasting characters chosen by Mendel were shape and color of seeds, round yellow seeds and wrinkled green seeds. First point, Mendel first crossed pure breeding pea plant having round yellow seed with pure breeding pea plant having wrinkled green seed and found that only round yellow seeds were produced in the first generation F1. Second point, from this it was concluded that round shape and yellow color of the seed were dominant traits over the wrinkled shape and green color of the seeds. Third point, when the hybrids of F1 generation pea plant having round yellow seeds were cross-bred by self-pollination, then 4 types of seeds having different combinations of shape and color were obtained in second generation of F2 generation. They were round yellow, round green, wrinkled yellow and wrinkled green seeds. Fourth point, the ratio of each phenotype or appearance of seed in the F2 generation is 9:3:3:1. This is known as the dihybrid ratio. Then fifth point, from the above result, it can be concluded that the factors for each character or trait remain independent and maintain their identity in the gametes. The factors are independent to each other and pass to the offsprings through gametes. Results of a dihybrid cross. Mendel got the following results from his dihybrid cross. First point, four types of plants. A dihybrid cross produces four types of F2 offspring in the ratio of 9 with two dominant traits, 3 with one dominant trait and one recessive trait, 3 with another dominant trait and another recessive trait and one with two recessive traits. Second point, new combination. Two new combinations of traits with round green and wrinkled yellow had appeared in the dihybrid cross F2 generation. P. Herons between dihybrid of monohybrid uses. First point, dihybrid cross is different from a monohybrid cross because it involves inheritance of two pair of contrasting characters. In monohybrid cross, only inheritance of one pair of contrasting character is studied. Second point, further result of monohybrid cross is based on two alleles of gene. Third point, results of dihybrid cross is based on two different genus controlling two different characters and their alleles. Second question, how is the structure of DNA organized? What is the biological significance of DNA? Answer, structure of DNA. The most widely accepted model of DNA is the double helical structure of James Watson and Francis Crick. Chemical composition of DNA molecule. DNA is a large molecule consisting of millions of nucleotides. Hence, it is also called as polynucleotide. Each nucleotide consists of three components. First point, a sugar molecules, deoxyribose sugar. Second point, a nitrogenous base. There are two types of nitrogenous bases in DNA. They are option A, purinase, adenine and guanine. Option B, pyrimidines, cytosine and thymine. Nucleoside and nucleotide. Nucleoside is equal to nitrogen base plus sugar. The nucleotide is equal to nucleoside plus phosphate. The nucleotides are formed according to the purines and pyrimidines present in them. Watson and Crick model of DNA. First point, DNA molecules consist of two polynucleotide chains. Second point, these chains form a double helix structure with two strands which run anti-parallel to one another. Third point, nitrogenous base. In the center of linker to sugar phosphate units which form a backbone of the DNA. Fourth point, pairing between the nitrogenous base is very specific and solves between purine and pyrimidines linked by hydrogen bonds. Then adenine links thymine T with the two hydrogen bonds, A is equal to T. Then cytosine C links guanine G with 3 hydrogen bonds, C is equal to G. This is called a complementary base pairing. Fifth point, hydrogen bonds between the nitrogenous base make the DNA molecule stable. Sixth point, each turn of double helix is 34A degree 3.4 nm. There are 10 base pairs in a complete turn. Then seventh point, the nucleotides in a helix are joined together by phosphodiester bonds. Significance of DNA. First point, it is responsible for the transmission of hereditary information from one generation to next generation. Second point, it contains information required for the formation of proteins. Third point, it controls the developmental process and life activities of an organism. Third question, the sex of the newborn child is a matter of chance and neither of the parents may be considered responsible for it. What would be the possible fusion of gametes to determine the sex of the child? Answer. First point, human beings have 23 pairs of chromosomes, out of which 22 pairs are autosomes and one pair, 23rd pair is the sex chromosome. Second point, the female gametes or the egg formed are similar in their chromosome type 22 plus X. Therefore, human females are homogametic. Third point, the male gametes or sperm produced are of two types. Fourth point, the sperm bearing 22 plus X chromosomes and the sperm bearing 22 plus Y chromosome, the human male are called heterogametic. Fifth point, it is a chance of probability as to which type of sperm fuses with the egg. Then sixth point, if the egg X is fused by the X bearing sperm and X's individual female is produced. Then seventh point, if the egg X is fused by Y bearing sperm and XY individual male is produced. Eighth point, the sperm produced by the father determines the sex of the child. The mother is not responsible in determining the sex of the child. Eighth Roman letter, higher order thinking skills. First one, flowers of the garden P are bisexual and self-pollinator. Therefore, it is difficult to perform hybridization experiment by crossing a particular pistil, which is the specific pollen grains. How Mendel made it possible in his monohybrid and dihybrid crosses. Mendel selected the plants to be used as a male and female parent. He removed the stamen from the flower of the plant, which was taken up as a female parent. Self-pollination will not be possible in this flow. Further, he also kept the stigma covered so that no other pollen other than the desired variety will fall on the stigma. The Mendel was above 40% self-pollination in the plants selected for crossing. Second question, purebred tall pea plants are first crossed with purebred dwarf pea plant. The pea plants obtained in F1 generation are then crossbred to produce F2 generation of a pea plants. Option A, what do the plants of F1 generation look like? Then option B, what is the ratio of tall plants to dwarf plants in F2 generation? Option C, which type of plant were missing in F1 generation but reappeared in the F2 generation? Option A, the plants of the F1 generation will be tall. Option B, the ratio of tall dwarf plants phenotypic ratio will be 3:1 in the F2 generation. Then option C, the dwarf plants were missing in the F1 generation but reappearing in F2 generation. Third question, Kavita gave birth to a female baby. Her family member says that she can give birth to only female babies because of her family history. In the statement given by her family members true, justify your answer. No, the statement given by her family members is false. The birth of male or female baby is by random combination of a sperm and egg. Sperm are of two kinds based on sex chromosome, the X chromosome and Y chromosomes. The eggs always have only X chromosomes, the sex chromosomes. In a sperm with X chromosome fuses with an egg, a girl baby is born. If a sperm with Y chromosome fuses with an egg, a boy baby is born. The sex of the baby depends on the type of sperm which fertilized the ovum. Ninth Roman letter, value based questions. First one, under which condition does the law of independent assortment hold good and why? Law of independent assortment. The law was proposed based on dihybrid cross. The cross involves the inheritance of two pairs of contrasting characters at the same time. First point, each character is determined by a pair of alleles, dominant and recessive. Second point, each allele is contributed by a parent to the offspring. Third point, the result of the dihybrid cross performed by Mendel showed four combination of characters in the ratio of 9:3:3:1. Fourth point, it can be concluded that the factors for each character or trait remain independent and maintain their identity in the gametes. The factors are independent to each other and pass off the offspring through gametes. Therefore, the conditions for the law of independent assortment are option A. Each trait is determined by a pair of alleles. Option B. The inheritance of all alleles governing the two traits must be independent of each other.